Okay, let's get started. Appreciate everyone being here and competing with the sun. Of course, it's probably all about the clicker questions, and as soon as I ask them, everyone will leave. But you'd be missing out on some of the coolest viruses we're going to talk about all term. These are the giant viruses. The coolest, of course, we're going to talk about on Monday, because those are the archaeoviruses that we work with in my lab. And then on Wednesday, Luis Ruedas is actually going to come and tell us a little bit about virus ecology and cute little mice, which we shouldn't really think of as being cute. And I'll give you one guess why. What kind of class is this? Virology class. So that's what's happening next week. And then, of course, Friday will be a review. Week from Tuesday at 8 a.m. will be the final. Yes, what everybody's all happy about. How many of you have filled out your online evaluations? Any of you? Yes, you all will by the end of the term? Yes. How many of you are coming to graduation, which is much more important? Yes, that should be a lot of fun. So I think those are all the things that I had to... Oh, the one other thing, see, I keep forgetting these things, is next Thursday, when you're going to need a study break anyway at noon, Klaus Frew from OHSU will be talking about cytomegaloviruses and how amazingly cool they are in terms of herpes viruses and why they make a great vaccine platform. So that's, I think, everything that's happening next week, which is dead week, right? You know, quote unquote. The regular biology seminar, yeah, next Thursday. Okay, so the reason you're here and not out in the sun is our clicker question. First one anyway for today. The sequence structures at the ends of the vaccinia-virus genome are most similar to the ends of the genome of papillomaviruses, polyomaviruses, adenoviruses, coronaviruses, flaviviruses. $15. 10. 4, 3, 2, 1. No, you're not going to have more time. So this is very similar, again, to the kinds of questions that I would expect to see on a final. There'll be sort of these compare and contrast questions. They will also be questions that are not necessarily in the notes, so you can go back and flip through them and find them immediately. So, of course, and open everyone. No, unfortunately not. So, what do people think? That it's adenoviruses, because that's what I said, right? Stedman said. If Stedman said, that's what the answer is going to be. But why did Stedman say that? Now, it's true to the best of Stedman's knowledge, which is a slightly different thing. It's the inverted terminal repeat thing. So I need recordings of you guys, actually, when you're chatting about this, and then also the hand movements here. Why not papilloma or polyomaviruses? Do these guys even have ends? They're circular. So they are all end or no end. Coronaviruses, what kind of ends do they have? Boring messenger RNA ends, who cares? And flaviviruses similarly. Okay, so we need to select the right answer here. And our next question is, the smallpox vaccine has to be administered X number of times or time to be effective. 1, 2, 3, 4, 5. That was 19.
Didn't they do it 42 million times?
Somebody's clicker making a desperate bid for freedom. I'm not seeing any more votes. We don't need any more time. No, we're happy with this. Okay, we'll give people time to show up. 4, 3, 2, 1, lift off. And we have one person who thinks 5. Everyone else thinks 1. And the main reason for a question like this is just to remind you that stuff that's in videos could be on exams as well, because I didn't mention this in class at all, but they kind of beat on it in the video. So yes, the answer is A. Hallelujah. Why is it not going green? There we go. So enjoy the sunshine. And we'll talk about giant viruses. Depending on how the extra technology works, we may or may not finish a little early. This will be the weekend. And so you can appreciate this big, nasty yellow thing in the sky a little bit more. So the giant viruses, gee, go figure. They're big. And in fact, some of them ridiculously enormous. And we'll talk about some towards the end of today's lecture that are even bigger than the ones that they talk about in the textbook. One of the authors, I don't know if you've read this chapter in the textbook, I think it's one of the more amusing ones. I think they do a really nice job of going through it. One of the authors, William Wilson, goes by Willie Wilson. That's his choice. So Scottish fellow working in southern England. Really hilarious. He did a great radio lab a few years ago talking about this whole process of the algal viruses versus the algae. And one of the things I forgot to bring with me, there's this really fun app that a group out of the University of Nebraska put together on viral wars. And so it's a comic, basically a graphic novel of the Aemiliana huxleyi virus against the Aemiliana huxleyi. And so I'll try and put that link up. Sorry, I was going to bring it, but didn't manage to do so today, so we may get out a little bit early. These, even though they're absolutely enormous, at least most of them, with the exceptions of the ones we'll talk about right at the end, are icosahedral. So even though these things are ridiculously huge, they still have that same icosahedral-like symmetry that we talked about way back when at the very beginning of the course. So now how do you get a smallest surface area, simplest structure, largest genome, say smallest surface area, largest volume is a sphere. And so how do you get close enough to a sphere? It's icosahedral symmetry. Some of these giant viruses actually have to do with climate. So viruses change the weather, which is really pretty amazing. Also are really important for the air we breathe. Not surprisingly, they're big viruses. They also have really big genomes. And then it always brings up this question is what is a virus? Getting way back to our definitions that we had right at the beginning. These things are certainly not small by any stretch of the imagination. They are obligate intracellular parasites. So that still is okay. They're still using most of the translational apparatus. But they also have a lot of the translation apparatus that they bring with them, which is really pretty impressive. A couple of key concepts here. NCLDVs, one of the worst acronyms I have ever heard of. These are the nucleocytoplasmic large DNA viruses. I like giant viruses way better. But you will see this in the literature. NCLDVs just rolls off the tongue, doesn't it? Is what a lot of people will call these things. these are extremely abundant viruses. So a lot of those dots that we talked about way back when, lecture 1, many of those are probably some of these giant viruses. If you look in seawater, there are very, very large numbers of them. A lot of them have internal lipid layers. So we'll talk more about some of the internal lipid layers on Monday, because some archaeal viruses also have these internal lipid layers, but it's a real protein capsid on the outside with a lipid underneath that. And then underneath that is usually the genome and sometimes even an extranucleocapsid protein. And then how some of them escape, best known for the Mimivirus is this Stargate structure, which is a really amazing change at one of the 5-fold axes of symmetry of these viruses, virions I should say, where they're able to get out. Large nucleocytoplasmic DNA viruses. Let's see, this should actually be nucleocytoplasmic large DNA viruses. You can tell I can't even get the acronyms right. These are a whole bunch of different viruses, and this is what we're going to talk about today. This is, together probably with the lecture on Monday, one of those lectures that we could easily spread into weeks and weeks and weeks of talking about these amazingly cool viruses. But this is really an overview course on these viruses. So PhycoDNAviruses, these are probably the best understood of these. These are the classic algal viruses. So algae, and this is eukaryotic algae viruses that are being infected by these. And again, not surprisingly, what do we have in the middle here? That's Stedman's put in uppercase. They're double-stranded DNA viruses. The Coccolithoviruses we'll talk a little bit more about. These are those that are potentially, and actually probably are at least locally, changing the climate a little bit. These also have what I like to call global plaques, so plaques that you can see from outer space. So you have the Earth and then a plaque you can see where you have virus infections, so you can actually see these from space. We'll look at those in just a couple minutes now. The Mimivirus, which at the time that the chapter in the book was written was the largest of the virions out there. Now we've got a number of larger virions, the Megavirus, followed by Pandoravirus and Pithovirus. And then some of these viruses even have virophage, which infect them. And so they're so big, they have viruses that infect the viruses, which doesn't really make sense if you just think about viruses as virions. But if you start to think about viruses as through their whole viral life cycle, then it makes a lot more sense that you'd have these, which are in there. So we'll start out talking about the PhycoDNAviruses. These are the algal DNA viruses. Algae are way more diverse than probably most animals, with potentially the exception of the insects in terms of number of species. So they're really a ridiculously large number of these things. They look pretty similar to each other. For the most part, you've got small little green ones, but you also have things like kelp, which is also an alga. So some of these are really large as well. About 50% of the world's oxygen is produced through photosynthesis that happens in these algae, and probably 20% of them are killed every day by virus infection. So it gives you a bit of an idea of the turnover of the algae, and then, of course, really critical for the air we breathe. And part of the, again, Willie Wilson's idea of humor is that without most human viruses, we might be okay. Without algal viruses, we'd all be dead. So that's his take on sort of these things. And again, I look to see if I've got the, is that NCLDVs? No, N. These are the large nuclear cytoplasmic DNA viruses, right? So again, if I can't get the acronym right, I'm not expecting you guys to remember it. But basically, all of these viruses are clearly related to each other, including the pox viruses that we talked about last time. So it looks as if you do phylogenies, so try and reconstruct how these viruses are related to each other based on their genome sequence. Turns out that there are a number of conserved genes, about 30 of them, which are present in every single one of these giant viruses. And those are all clearly related to each other to the exclusion of any other viruses or any other cell types. So they probably came from some kind of common ancestor back here. So pox viruses, baculoviruses that we didn't talk about, these are insect viruses. There's a nice chapter in the book about them. Iridoviruses, which are also giant icosahedral viruses. These are ones that infect mostly insects. And then we're mostly going to talk about the PhycoDNAviruses here. These giant viruses of algae. And more or less to scale are these Mimiviruses here. So you'll see these are considerably larger than any of these. And remember, this is a M, not an N, so it's not a mini-virus, but a Mimivirus. So the best studied of these PhycoDNAviruses, partly because this is a picture of my lab down here in the corner. We even have studied these guys because they're really big. These are the Paramecium Berseria chlorella virus. Paramecia, these are what kids look at under their cheap microscopes. In fact, my 10-year-old, we're going to be looking at this next Friday in her class in 5th grade, looking at some of these paramecia. These paramecia very often have these chlorella alga, which live inside them. And these are a beautiful green color. And basically, the alga, which are inside the paramecium, cause these paramecia to be green and allow them basically to photosynthesize. They take up all the photosynthates, which are made by these algae, and then can grow. One of the reasons that people are understanding these viruses and are really excited about it, these chlorella can actually grow outside the paramecium as well. You can grow them on plates, and this is a plate right here. Each of these lighter green spots represents a plaque. So this is a pretty classic plaque assay. And so the group mostly of Jim Van Etten at the University of Nebraska, where they produce the graphic novels, has been studying these viruses probably the longest of anyone. And the main reason he's been studying this is because you can basically pretend that these chlorella are bacteria. You plate them as lawns, you do really regular plaque assays, you can count, you can follow what's going on with each of these individual viruses, and even I can grow them, so they can't be too hard to work with. The first couple of surprises, first, these were giant viruses, but the second was looking at the genomes of these viruses. So the genome of PBCV1, this is from their paper from, excuse me, the Van Etten lab. I like this one a lot better because it breaks down all of the different kinds of genes that were found in this almost 300,000 base pair length genome. How big are the poxvirus genomes? About 200,000. So these are another 50% bigger in terms of the genome. But in terms of genome structure, they're very similar to the poxviruses. They're linear, double-stranded. They've got these covalently closed hairpin ends and long inverted terminal repeats. So they're probably replicating in a very similar way. That's still a little bit up in the air. There are a couple of really interesting things, however, when they sequenced this genome that they found. One was that there are 11 tRNAs that are encoded in the virus genome. Like, wow, I thought they were always using cellular translation, right? Well, yes, but in this case, probably the virus genome has a lot of genes which have a particular codon that they use, that then the host doesn't have as much of the tRNA, doesn't use as much of that codon, so they need more copies of some of those tRNA genes. And if you do an analysis of the coding sequences in the genome here, that's exactly what you see. There's also a ubiquitin gene that's encoded in this virus genome. Whether it came from the algae that were being infected, some kind of horizontal gene transfer, is not entirely clear. There's a bunch of transporters. There's even a self-splicing intron, which is present in this genome. And Dr. Lehman here in the chemistry department, in fact, worked on that a little bit. The reason I like this figure so much is it breaks down a lot of the different kinds of genes which are present here. I know this is too small to read, That's on purpose. The major categories here, however, are metabolism, DNA replication, recombination, and repair, bunch of genes, lots of nucleotide metabolism genes, transcription genes, protein synthesis genes, including all those tRNAs, which are down here at the bottom, cell wall degrading enzymes, sugar and lipid metabolism. There's a bunch of genes in these viruses, and we'll see in most of the other algal viruses as well, which are important for changing the metabolism of the host. So once you have an infection, you've changed the metabolism of the host in such a way that it makes it actually better for producing more virions. And so this process is very well understood in these cases. Signaling, DNA restriction and modification enzymes, a whole bunch of DNA methylases are present in these genomes. Integration and transposition, got transposons, got, excuse me, also topoisomerases, and then miscellaneous and structure proteins as well. What do these guys look like? They are icosahedrally symmetric in a somewhat bizarre way. And the way that is they've got these, what they call trisymmetrons. So basically, it's a icosahedrally symmetric particle. But a lot of the hexamers, if you think about this again in terms of your regular icosahedron, are organized into these triangular faces, which they call trisymmetrons. And then those flank these areas of five-fold symmetry, which you can see right here. So that's a slightly different way of putting together the whole structure. However, the major capsid protein, this VP54 protein, has an extremely similar structure to a number of other viruses that we've already talked about, most specifically adenovirus, which has a double beta barrel structure. There are bacteriophage that have this double beta barrel structure, and as we'll talk about on Monday, and I talked about earlier on, the STIV virus and archaeovirus also has this double beta barrel structure on the outside of the virion, the major capsid protein. This fits together, in this case, as a trimer of proteins that gives you your hexameric subunit, which you can then fit together to make this icosahedral structure. These proteins are heavily glycosylated. That's what these yellow dots here represent. Glycosylation is all of the sugar residues that get added on the outside. All the glycosylation we've talked about so far, because a lot of viral proteins are glycosylated on the outside, that's all cellular glycosylation. Here, it looks as if all that glycosylation is driven by viral proteins. So probably this doesn't need to go through the Golgi process, doesn't have the normal glycosylation process. Probably all the glycosylation is happening through the activity of viral proteins that are encoded here. Originally, when people looked at this, they first thought it was a nice symmetrical icosahedron, but then they did more cryo-electron microscopy and they found that it had this spike at one of the vertices, which should seem really familiar based on herpes virus, which has a one specific vertex, but of course most of the bacteriophage have this one specific vertex. Not surprisingly, it seems to be this one specific vertex which interacts with the cell. And you can see it here. Here's one of the virions just about to interact with the cell. Here then, it's clearly interacting with the cell. These are algal cells. They're usually either inside the paramecium with PBCV1, but can also survive outside the cell. So they've got pretty thick cell walls. Unlike the case for all the plant viruses, where you have to have physical degradation of this cell wall somehow, either aphids or some other way of getting degradation, these guys have enzymes that are in fact connected to that tip structure. So those enzymes break down the cell wall. And they break down the cell wall so that you can get the internal membrane here to fuse with the plasma membrane, which is present here in these cells. So you break down the cell wall, Underneath the cell wall is the plasma membrane that then fuses with the internal membrane of this virion. Once that's fused, the genome contents come spilling out. You still see the rest of the capsid on the outside of the cell, which really looks a lot like a bacteriophage. They've bound, their genome is released, that's it. When you have this fusion event, you have depolarization of the membrane. People, in fact, have used this to study membrane depolarization, looking at what happens when you have membrane depolarization excuse me, in a lot of these algae. Once you have release of the genome here, these are double-stranded DNA viruses. They're infecting algae. These are the eukaryotic algae, which then have, of course, nuclei. These nucleocapsid proteins, then, which are released, get transported to the nucleus, and then there are a bunch of viral proteins that degrade the host DNA. So a bunch of DNases that are present in the viral genome. Now, you remember I mentioned all those modification enzymes when we talked about the genome before? The genome of the virus is heavily methylated. The genome of the host is not. So these restriction modification enzymes are coming in with the virus, unlike the case in bacteria, where it's usually the host which can then protect against the virus infection. Now it's the virus which is breaking down the host DNA. Why would you want to break down host DNA? Provide nucleotides so you can make more virus genome. So it's a wonderful source of nucleotides. Once you've gotten all these nucleotides, you replicate this ginormous genome, probably through a rolling hairpin mechanism like we saw with the pox viruses. Then you start to put these viruses together. Remember, these are pretty big. So here's one of the chlorella here. Each of these are virions which are being assembled inside this infected cell. Eventually, there are so many of them that they burst the cell through cell lysis. These are now released, can go off and bind to another cell, cause an infection, and go through the whole process. This takes about 6 hours, and what I don't have here is the pictures of when we were growing chlorella in my lab. You start out with this nice green sort of sludge because you're looking at all of the chlorella. Once you add the virus, leave it for about 6 hours, that green all goes away, it all becomes yellow, and you can, even under the light microscope, see some of these poor chlorella that have completely lost their contents because all the viruses are being produced. You can't actually see the virions under the light microscope. These are electron micrographs. We'll see the Mimiviruses, which you can see under the light microscope, in just a second. I'll switch gears a little bit and talk about the Coccolithoviruses. These are also algoviruses, but these are algoviruses which are infecting Coccolithophores. And I love this picture here because this is a classic Coccolithophore. It's got these calcium carbonate shells around the outside, and there's a virion right there attached to one of these Coccolithophores. How these virions get through this really amazing cage on the outside is still a pretty open question. There's some discussion about that in the text that I'm not going to get into, having to do with the sexual state of how these guys replicate. They can replicate asexually or sexually, and maybe that the virus can infect one of those states and not the other one. What I mentioned before were these plaques that you can see from space. This is just off the coast of southwestern England. Portsmouth is right about there. That's where Willie Wilson's lab is. So that's why he's quite excited about this. But all of this white stuff that you see here, that's from these Coccolithophores, but the Coccolithophores after they've been lysed. If they're not lysed, you see nothing. If they're lysed, you see this huge amount of these plates, these calcium carbonate plates, which just basically fills up the ocean. And you can see these plaques from space. Now, it's not one virus which is there, but it's a virus infection. So you can see these virus infections from space. And a little bit further over here, off about where the exit sign would be, is Dover in England. What's Dover famous for? The White Cliffs. Where do those White Cliffs come from? From all of these Coccolithophores, which have then been broken apart because of virus infection. So the White Cliffs are due to virus infection. So we've got this plaque that you can see from space, and also geological records of virus infection that are, you can see these virus infections, or what is left then from a lot of these virus infections. The other thing that these Coccolithoviruses do when they infect and lyse EhuxV86 is they release this chemical called dimethyl sulfoxide. That then is a gas, gets up into the atmosphere, becomes a sulfate. These sulfate particles become condensation for cloud formation, which then will cause less sunlight and then in some cases actually rain as well. So what happens, you have a big bloom of EhuxV86 where you have huge amounts of cells which are being produced. This is before the viruses are lysing them. How are you getting this huge bloom? Well, these are photosynthesizing algae, right? So lots of sunshine. So lots of sunshine, you get large production of these Coccolithophores. The viruses then kill them off, release DMS, and you end up with cloud formation, which is then probably going to block any more of these massive blooms because it's a lot less solar radiation. So how you could have this in terms of a selective mechanism, I think, is a really open question. And they also mention Gaia hypotheses and things like this as well. But certainly, this process, the viruses lysing EhuxV86, and producing DMS, means that there is less of this HuxV86 around. It may be just a coincidence, and again, whether there's any selection which is going on there is really an open question. But certainly, the viruses kill off the EhuxV86 when they have a big bloom, and also indirectly are stopping future blooms that are happening. So that's what happens on a global scale. People, of course, are also interested in what the genome looks like of these viruses. This is the EhuxV86. Tells you also there are a whole bunch of different viruses that are infecting Emiliana huxleyi. Emiliana huxleyi, by the way, is that particular Coccolithophore that we looked at with the beautiful sort of shield-like plates around the outside. There are lots of different Coccolithophores which are there, each of which has their own separate viruses. EhuxV86 is one of the most common of these. So this EhuxV86 genome clearly encodes its own RNA polymerase, which should sound really familiar based on what kinds of viruses that we talked about on Wednesday? Pox viruses also encode their own RNA polymerases, but 80% of the genome here, we have no idea what it does. So another example of where a lot of the people call dark matter, I'll call dark matter of sequence space, is present in a lot of these viral genomes. But some of the more important genes that have been able to be recognized, which are these that are colored in something other than green, here are important for ceramide biosynthesis. So what the heck is ceramide? Ceramide is another one of these glycosylation products, very specific sugars. All of the enzymes for making ceramide are present in the viral genome. Normally, EHUX does not make ceramide. If it's infected with the virus, then it does make ceramide. Why? Well, it turns out that ceramide is really important for regulating apoptosis. So the production of ceramide leads to apoptosis, and in this case, it appears that apoptosis is what the virus wants in order to be able to get out of the cells. And in fact, it may well be apoptosis which is leading to the cells degrading and all of those plates coming off, forming the viral plaque, and eventually the cliffs of Dover. So the virus is regulating when the cell undergoes apoptosis by making this ceramide. One of the other interesting things about the ceramide here, also called a glycosphingolipid, these also have a lipid attachment to them. It's a very specific sphingolipid which is made just by virus-infected cells. And so what you can do, and people have actually done this, is take a research vessel, a ship, and basically sail across the Atlantic, taking samples every once in a while, and just by looking for this one particular ceramide sphingolipid, see where you have virus infections. And so you can literally count the amount of virus infections that are there just by looking at this one particular glycosphingolipid. And we're really excited about this because this might actually be what we call a biosignature, which is then, again, it's very specific. You only see it from a virus-infected cell. It's evidence that a virus has been there. Turns out that lipids are also really good at being preserved in the geologic record. And so people have now actually gone back to the White Cliffs of Dover and found some of these sphingolipids in some of the rock structure. And so you can also start to think about really old viruses by looking at these biosignatures. Or, as some of you may have noticed, the NASA just decided on what instruments they're going to put on the spacecraft they're sending to Europa. They're not actually looking for glycosphingolipids, but maybe the next mission they will be.
So is that the ceramide biosynthesis, is that coded in the algal genome?
No, so this is in the viral genome. So it's a very specific coding sequence in the viral genome. And there are other mechanisms for regulating apoptosis in the algal genome, but they're not dependent on ceramide. I mean, again, we talked about the climate modifications that you have here, again, because of the production of DMS. The thing to be worried about in terms of climate change is a lot of you may have heard about ocean acidification. I think ocean acidification is potentially much more of an issue than the change in temperature. And the reason for that is that calcium carbonate, the thing that's making those plaques and making the nice structures on the outside of these coccolithophores, is very dependent on pH. And if the pH of the oceans goes down too much, we're not going to be able, i.e. these coccolithophores, are not going to be able to make those plates anymore. If you can't make the plate anymore, you can't protect your alga. What are those algae doing? Producing oxygen. So there are a number of predictions about the oxygen levels on Earth, what they'll do as the ocean acidifies. And some of them are pretty dire predictions in terms of looking at that. So even if you don't live in a low-lying area near coasts, you're certainly breathing. So that's an interesting thing to think about. You know, make everybody happy for the weekend. So finish out talking about the Mimiviruses and then some of the relatives of the Mimiviruses right towards the end here. This is basically just to show that these things are ginormous. These bars here, this is 200 nanometers. These guys are almost a micron across in terms of their size. Nice icosahedral structure here with these long tails on the outside. These are the really big viruses. This is a light micrograph. Each of these little dots in here represents one of the virions. So you can literally see these guys under the light microscope. I've yet to do this. I really want to do this sometime. First time I listened to a talk, one of the guys who was working on these, he actually showed one of these micrographs. I was like, wow, you can see viruses under the light microscope. So they're really big. Some people call them gyruses, so the giant viruses. I don't particularly like this terminology. But again, they're visible in the light microscope, 750 nanometers with the hairs on the outside. All of these giant viruses so far have been found infecting amoeba. And so this is a thin section of an amoebal cell down here, in fact, from giantvirus.org. Lots of fun pictures of giant viruses there. Here's a Mimivirus inside an amoeba, which thinks it's getting some food, but unfortunately, they're not going to be very happy, that particular amoeba here. Where were these originally found? They were originally found in cooling towers, a place called Bradford, England. They called them Bradfordococcus for a long time because there were these bacteria and looked as if they were growing intracellularly. And in fact, the first people to show that these were viruses at all were people who were working on rickettsia. And rickettsia is an obligate intracellular bacterial pathogen. And so the group in England who'd been working on this Bradfordococcus sent the group in Marseille, in fact, two different groups that are now competing with each other, apparently can't even stand each other enough that they don't even go to the same meetings or talk to each other. Different story. The French, you know, very strange. I know both of them. They're really great people. So they were the ones who showed literally 11 years later that these were in fact viruses, and they went through a really classic viral replication process. And once they looked at them under the electron microscope, now, anytime I would look at something like this, I'd say, hey, you know, that doesn't look like a bacterium. Bacteria don't have these nice icosahedral structures. They don't have corners on them. And so that was basically the mechanism that they used to find them, and then eventually showed that they replicated only through this whole infection process. Of course, they also sequenced the genome. Turns out this genome is over 1 million base pairs in size. So we went from 200,000 in smallpox to 300,000 in PBCV1 to over 1 million base pairs. This is bigger than a lot of bacterial genomes. Well, not a lot of bacteria, but some bacterial genomes. Almost all of the genome sequence is coding. Again, not unlike bacteria. It codes an RNA polymerase, a bunch of DNA repair enzymes, but most surprisingly were all the translation enzymes which were encoded for in this Mimivirus genome. Aminoacyl tRNA synthetases. I think, if I remember correctly, there are 4 different aminoacyl tRNA synthetases. So not just tRNAs, which it also encodes, but aminoacyl tRNA synthetases. So you make the tRNA and you have the aminoacyl tRNA synthetase that will bind to that, put the appropriate amino acid on it, check and make sure if it's the right one. So really the whole quality control mechanism. They also encode cap binding proteins, shown here. This is in review. Cap binding proteins, no other virus genome to date have been known to encode these cap binding proteins, and also a number of homologs of some of these initiation factors as well. So no, they look really hard. There's not a ribosome that's encoded in these genomes, but we're getting really, really close to looking at this. It looks as if a lot of this genome, instead of being horizontally transferred, so one of the questions was maybe Mimivirus is just really good at pulling in other genes from its host. There was a whole lot of discussion in the literature about 3 or 4 years ago, maybe more like 5 or 6 years ago now, where people were arguing about whether Mimivirus had stolen all the genes from amoeba or if they were really viral genes. And most people agree that they're really viral genes here. And again, if you look at those genes that are conserved in the nucleosarcoplasmic large DNA viruses, then you see that these genes, those conserved genes, really nicely put these viruses related to all these other giant viruses, including things like smallpox. And a lot of this genome here actually seems to have been made so big through duplications of various different genome fragments. Even though this is a ridiculously large structure, at least as far as a virion is concerned, it's clearly icosahedral, and the T number is a pseudo 1,179. I don't expect you to go and calculate the number of particles or anything like that, and I don't expect you to remember this number, but it's really clearly almost quasi-equivalent structure, the way that it's fit together. Really pretty amazing. It's got 2 lipid bilayers inside this capsid, and then, of course, all of these fibers on the outside. This is one of those French groups, Jean-Michel Claverie, where you can see this individual virion with all these fibers on the outside. Why are the fibers there? Probably to confuse the amoeba into thinking that these things are food. And a lot of amoeba eat bacteria, and this is the reason that Mimivirus has that name, because it's thought to be mimicking bacteria. So that's where the mimi comes from, is the mimicking of the bacteria. So you've got the fibers that are on the outside, this pseudo T equals 1,179 icosahedral structure. How do you get the genome out of this? Well, this was another big surprise in terms of studying the Mimiviruses, was that they have this structure which was called the Stargate in terms of being able to release the genome. And that is that one of the five-fold axes of symmetry of this pseudo T equals 1,179 actually has a different structure. And that different structure here then appears to be how these viruses are releasing their genome from those vacuoles. Again, you saw all the pictures you see of the Mimivirus are inside the amoeba, where the amoeba is basically trying to eat them. Well, they escape then through this Stargate structure, and this is a scanning electron micrograph of this, and you can actually see these in the scanning electron microscope because they're so huge. And this is the nucleocapsid here on the inside. Anybody recognize what this is? Alien? Yes, exactly, from old science fiction movies. So they obviously knew what was happening here. Way before, actually, that movie was well before Mimivirus was actually discovered. So let's see if we can go to this link, see if this one will actually work, because it's a lot nicer to see the Mimivirus structure here. This is just an animation rotating the Mimivirus. Here's that Stargate structure. Normal five-fold axes of symmetry, and again, just one which has this really specific structure which is present on the outside there. Again, here's coming around to this particular Stargate structure, and that's, again, been quite well characterized, all of the different stages of that opening. So when you're in there, that's what happens. Let's stop this right now before we get the next one. Yeah?
What's the mechanism that opens the Stargate?
What's the mechanism that opens the Stargate? Let's see. Wasn't it actually lining everything up properly in the bad movie, but no, in the film product? No. So it's not entirely clear what the fusion process is, how the changes happen in these are what they call the digestive vacuoles then of the amoeba, and how that fusion takes place. It's not an obvious sort of conformational change, pH, etc. So yeah, how the Stargate opens is not obvious. Again, maybe we should, all sci-fi stuff in terms of how that's actually working. So that's the Stargate process. You get, once the Stargate forms, you release the genome here. That then genome goes to a particular place in the amoeba, which people then call the virus factory. It's where all of the viral proteins are. These guys also degrade the host DNA and use that DNA to make more of these virions. That process between engulfment and actual lysis of the cell only takes about 12 hours. So it's a really fast process where you have infection, Stargate opening, formation of this viral factory, and you can in fact see some of these viral factories here. Just as you stain with nucleic acid binding stain, that's up here in E, the nucleus looks like this. The viral factory has even more DNA in it now than the nucleus does. And again, these are 1 megabase pair genomes. They're pretty big. And just completely takes over the cell. These guys are produced, lysed, and move forward. So this is the classic replication process for the Mimiviruses, and as I think Willie, probably I'm sure it's Willie's text in the chapter, say, well, my virus is bigger than your virus, was always the next step. So soon after, actually a couple of years after the Mimivirus, then we had the Megavirus. This was 2011. Slightly bigger genome, 1.2 million base pairs, 7 amino acyl tRNA synthetases, a bunch of genes that were not homologs to Mimivirus, and I think we'll actually skip this YouTube commentary, in fact, by Jean-Michel Claverie about how absolutely wonderful he is and this giant virus. But that wasn't big enough. We then found the Pandoraviruses, we being again Jean-Michel Claverie's group. These guys are ridiculous. So 200 nanometers right here. Remember the Mimivirus on this scale would be about half of one of these things. These again are light micrographs of these viruses. Here on the inside is something they think might be a little bit like a nucleus, not entirely clear here. Then this is the plug-like structure here at the end. These are now no longer icosahedral. They're a box, they call them Pandora's box, or a little bit more like an amphora. These genomes are even bigger. This is beginning to be the same kind of size as some of the free-living bacteria. In fact, there's some free-living bacterial genomes that are less than 2 million nucleotides in terms of their size. This is about the same size as one of our favorite organisms, archaeal organism, Sulfolobus solfataricus. This is, again, very few sequences, very distant relative, of the Mimiviruses, these giant viruses. And then one of the things that Jean-Michel is trying to say here, most people don't agree with him, but that these are relics of what he calls the fourth domain of life. So we've got eukarya, we've got bacteria, we've got archaea, and he thinks that these are the remnants of a fourth domain of life. Again, most people don't agree with him, but yeah. Why did they only get discovered recently? Because they don't make humans sick. That's the basic reason. And who cares about viruses of amoeba? Yeah, why should you care about viruses of amoeba? And the only reason they actually cared about viruses of amoeba, the only reason they found these things in the first place, is they thought they were like rickettsia, which are causing diseases in humans. And in fact, that particular cooling towers are infamous for things like Legionnaire's disease, where you have bacterial infections that happen. And so that was what they were thinking. They were figuring it was disease. And then they found these giant viruses, and then they decided, hey, we're going to study these giant viruses. So that's probably why it's taken so long to find a lot of these things. And if anyone's done electron microscopy, you see blobs all the time in electron microscopy. Is that blob a legitimate blob and something which you have a whole bunch of them, like you have here? Does it actually replicate? As my postdoctoral advisor always used to say, einmal ist keinmal, once is never. So if you see a blob that looks really cool, unless you see a whole bunch of blobs that look really similar to each other, then you don't believe it. Yeah?
Did the Megavirus have hairs on it also?
Yes, the Megavirus also had hairs on it, on the outside. Pandora did not. And they were looking basically at the same place there. But, heck, that wasn't big enough, so we're going to find an even bigger virus. And this was last year, March of 2014, the Pithovirus, which is over a micron in diameter, the individual particles here. So herpes simplex virus for scale, HIV for scale, and the Pithovirus, which is right here. And what I did want to do is try and go to this Slate article here, where they talk about reviving this giant virus because this particular virus was isolated from permafrost. And it's the giant virus risk. I wouldn't push the whole risk thing too terribly here. I'm going to see if we can actually get the audio on this. No, it's not doing audio for us. Okay, I will let you check this out. The whole story is here about why global warming is such a horrible thing, and we're going to discover all these new viruses which are going to come out of the permafrost that infect the amoeba. Oh no. Have a good weekend. Enjoy the sun.